Hello, my name is Mauricio Rodriguez from Escuela de Biología at Universidad de Costa Rica, and I will present our research on genetic structure in birds with different levels of tolerance to urbanization, the case of the white-eared ground-sparrow and the House Wren. For some species, urban settlements may limit their movement, reducing the connectivity within the metapopulation. This may reduce gene flow, increasing inbreeding and genetic structure among subpopulations. However, for some species, connectivity increases in urban settlements because they can exploit the resources found in urban areas, and we can expect them to experience the opposite genetic effects. Identifying how urbanization affects genetic connectivity of wild animals like birds is important to determine the long-term resilience of these species to urbanization. In this study, we intend to compare the effects of Costa Rican urban areas on population genetics of two bird species with different levels of tolerance to urbanization. This is the case of the white-eared ground-sparrow, Melozone leucotis, found in dense thickets, coffee plantations, and secondary forest patches, and Troglodytes aedon, House Wren, found in open and semi-open areas, forest edges, and previous evidence suggests that this species has benefit from urban development. To study genetic structure in both species, we sampled 5 sites in Costa Rica: Monteverde, a sparsely populated area with coffee plantations and large forest patches connected to mature forest; Heredia, Universidad de Costa Rica, and Jardín Botánico Lancaster, located in the Central Valley of Costa Rica, an area characterized by a high density of populated and urbanized areas, with small secondary forest patches and coffee plantations found within; and finally, Corredores, where we only sampled Troglodytes aedon, and this last site is sparsely populated, characterized by intensive oil palm plantations. We captured the birds using mist nets and some playback, and collected blood from 53 individuals of Troglodytes and 56 individuals of Melozone. Then we did the DNA extraction and amplified 8 microsatellite loci on each species. We calculated general population genetic statistics using R, and samples were clustered using structure. We found significant genetic spatial structure in both species, but structure was considerably higher in Melozone than in Troglodytes. Supporting these results, structure software grouped samples of Melozone into 2 clusters, grouping Monteverde individuals into 1 cluster and Heredia, UCR, and JBL into a mixture of 2 clusters. Then, for Troglodytes, the structural analysis cannot accurately cluster individuals, suggesting that all Troglodytes belong to a single large pygmatic population. This contrast suggests a strong difference between the connectivity of both species, Melozone being more restricted and Troglodytes more connected. To test if this difference may be attributed to urbanization, we perform a circuit theory analysis of connectivity using Circuitscape, based on resistance values from land use and cover, assuming that urban areas provide the highest resistance. In Melozone, connectivity, shown in blue, is restricted to coffee plantations, forest areas, and semi-open areas among populations, and resistance increases along urbanized areas. Using this model, structure increases with resistance. For Troglodytes, a similar pattern is observed, suggesting that if urbanization impacts structure, Troglodytes should have a similar structure value as Melozone. However, this contradicts our observations, and Troglodytes has a lower structure. As we will see in the next slide, structure is also affected by environmental climatic conditions. We perform a niche modeling analysis using environmental variables based on the Maxent algorithm, and then we use the habitat suitability values as an input for another Circuitscape analysis. We encounter that Melozone is restricted to the Central Valley of Costa Rica, and its connectivity due to habitat suitability correlates with its structure. However, for Troglodytes, we found it to be distributed countrywide, and its low value of genetic structure among population explains the lack of correlation with climatic connectivity. These results evidence the negative effect of urbanization over the connectivity of one of the species, Melozone. As shown by the animation, during the last decades, its habitat has been lost and replaced by urban areas, especially since coffee plantations and thickets are not included in any of the conservation categories. This could drive the species to a diversity loss, which could ultimately lead to an extinction vortex. Urbanization will have a lasting impact on habitat availability of avian species, and further information on genetic structure will aid the development of conservation strategies. Thank you for listening. Goodbye.